last time?
We started with spermatic chain society, which is basically developing a pathway for sperms from primary removable germ cells. And it begins with puberty, and in embryo, the PTCs reach ridge, where the testes form from two leg structures called sex cords. And once the PTCs get to the site, they turn into gonocytes, which then enter the sex cords and have the ability to make sperms. And then when development of the testes is taking place, the sex cords form the seminiferous tubules. And when that change happens, BNP8 increases. And then gonocytes differentiate into another type of stem cell called spermatogonia. And spermatogonia change to type AS spermatogonia that are true stem cells.
Okay, then what happens?
So there are true stem cells, and we set an example if you were to destroy all the sperm in the adult male and have one type AS spermatogonia, it will repopulate the sperm population. And then we talked about the cross section of an embryonic seminiferous tubule, where you have epithelia, the lumen, you have type A spermatogonia, centrioles cells, linear cells, and blood vessels. The centrioles cells secrete glial cell derived neurotypic factor, GDNF, and stem cell factors. And then the linear cells secrete testosterone. The type AS spermatogonia niche is the combination of all of them together. And then
Okay, then what happens?
We talked about how adult male type A spermatogonia cells can produce 1,000 sperms per second. And then there's no meiosis in males until puberty. So then we talked about the spermatogenesis phases. So phase 1, the proliferation phase, phase 2, meiotic phase, and then phase 3, spermatogenesis. And then we started talking about how type AS spermatogonia can divide asymmetrically. And we talked about here, there's the leukoid mutation, which can occur here, and it results in sterile males. And the transcription factor for type B is affected.
Your leukocyte came very late. Before that, what happened to type A1 asymmetric vision?
So type AS can differentiate into type AS, which is the same as it was before, and type A1 spermatogonia. And then that will be used to produce sperm. So then mitosis occurs 5 times to produce type B spermatogonia, and it's called the symptom. The symptom, the one where
Syncytium.
Syncytium.
Yeah.
With the cytoplasmic bridge, which that is just a cell with multiple nuclei.
Okay, then what happens?
Then the type B spermatogonia undergoes mitosis again to form a primary spermatocyte, and then something in it changes, which is all meiosis, and the Sertoli cells secrete the GDNF and SCF. And as GDNF lowers and SCF increases, and with high STRA8, differentiatic division occurs, which creates the secondary spermatocytes. And then those
The
secondary.
retinoic acid come from where?
Huh?
RA come from where?
What comes from where?
Retinoic acid.
I said retinoic acid.
From where RA comes?
Oh, it comes from the Sertoli cells.
Yeah.
Sorry.
Yeah.
Which makes the high levels of STRA8. That causes the first meiotic division to produce secondary spermatocytes, and then the second division produces spermatids, which are technically haploid, even though they still have multiple nuclei.
Okay, then what happens? Were you able to go through the notes?
I got the notes last night. I haven't finished
Okay.
going through them yet.
Okay, then what happens?
Oh, me still, sorry.
Yeah.
So as these differentiate, they kind of move down towards the lumen. So the spermatids are at like the nearest point of the lumen compared to everything else. And then
Then we talk about Luxoid, right?
Yeah.
Yeah. So what is Luxoid mutation?
With which one?
The Luxoid mutation.
Luxoid.
why it is so? Actually, you're writing everything, because if that is the question in the exam, then okay, then what happens to look sorry?
So, my notes are kind of boring here, but it doesn't have the rat receptors for GNDF.
No.
That was,
Yeah, I didn't get
that was
that
something
thinking.
similar.
Yeah, what is
But the
Luxor?
Luxoid transcription factor for type B is affected. All the stem cells become sperm cells immediately, which means there's no more stem cells to make more sperm
Yeah.
throughout the life.
Yeah, so that is with the transcription factor mutation that maintains them as stem cells. So if that mutation is there, all those stem cells then will be changed to the type B and everything becomes spermated, spermated. So no more sperm, that's why there's children. So the GDNF, what was the GDNF story?
Is it the one with the ret receptor?
That was what the ret receptor
I
is.
didn't know that one either. I was
Yeah, so we said that the ret receptor mutation, ret is the receptor for the GDNF. GDNF is a trophic factor. We said that this GDNF is essential for many type of neurons, as well as we say that that is required here also, because the GDNF is required, right? So if there is a mutation in ret that cause multiple symptoms, one of the symptoms is Hirschsprung disease. We talk on the day one, right? Those guys don't have any neuron in the distal part of the gut.
What was that
Then-
name, sorry?
Hirschsprung disease. So if you have ret mutation, so they will have no neuron in the distal part of the gut. So they will have megacolon-like symptom, right? They will have no kidneys, because kidney development also is regulated by ret and GDNF signaling. They will have no neuron in the substation nigra, so they will be more like a Parkinson person, right? And of course, we are living there, important player in the spermatogenesis and also in the testis development, so they have a lot of problems here, right? So then we were on the last, everything else is clear, okay? Okay, then we were on the last, we said, there was a proliferative phase, we said there is a meiotic phase, and then we are saying the last part is spermiogenesis. Spermiogenesis. So spermiogenesis is essentially spermatids to sperm. Spermatids to sperm. So what is happening in this case, the first thing is going to happen is construction of acrosomal vesicle. Construction of acrosomal vesicle. Now, this construction of acrosomal vesicle is from Golgi, right? So what is going to happen, for example, just going to give you an example of normal cell. This is your spermatid, okay? And what you have here is, you have the regular Golgi structure, like they have this Golgi-like structure. Here is your nucleus. So this Golgi is going to change into a vesicle, and that is going to become like this. So this is your Golgi, and that Golgi is going to form a structure like this, and that structure will become acrosome vesicle. And right here is your nucleus, right? And remember, what was the structure? This was the structure like this. Right here is your spermatid. And this is the lumen of seminiferous tubule, right? So the sperm should come in this seminiferous tubule, right? So what is going to happen here? This nucleus is now formed, and what happened that right here is the centriole. From the centriole, this is centriole, and we know that there are two centrioles there, but from here, the formation of flagella takes place. This is formation of flagella. Now think about this, that here the acrosome is being formed, and the flagella is forming like this. This is the flagella, right? But this flagella is not in the lumen, right? To have it in lumen, you should have the rotation of the nucleus. So when the nucleus will rotate, the centriole which is here, that will also rotate. Okay? So what happened? So this is the centriole, this is the nucleus. So what happened? The rotation of nucleus takes place. So this is step 1. Step 2 is formation of flagella, and step 3 is rotation of nucleus. So this rotation of nucleus takes place, and what is going to happen then? So this is your spermatid again. This is your acrosome, and now your nucleus has rotated. So what happened? The nucleus is there, and because of that, this centriole is coming here, and this flagella is now in this direction. This flagellum is like this. So what is happening? Because this rotation has taken place, this flagella which was going on this direction, now this flagella, if I'm drawing it again, will be like in the lumen. Okay? So now this flagellum is now in the lumen of this. So far it is okay? And remember, they all are joined together. There is another, they're all joined together. So all the flagellum will be kind of in the. So far it is okay? Then what happened? The nucleus condense. Step 4, nucleus condense. And how that happened? The histones are replaced by proteamides. Histones are replaced by proteamides. Okay. Now proteamides are rich in arginine. Arginine is a basic amino acid, right? So in that protein, proteamides, 60% of the amino acids are actually arginine. Arginine are highly basic. So basic will have what charge? Positive or negative amino acid? Yeah. So arginine is highly basic. What happened? This arginine make it more condensed. So because of that, the chromatin is condensed. So chromatin is now highly condensed. And when the chromatin is condensed, that means transcription will be shut down or on? Shut down. So therefore, complete shutdown of transcription takes place. Complete shutdown of transcription takes place. So chromatin, nuclear, that means that nucleus essentially condensed. Okay? Very tightly packed chromatin is there. Very condensed chromatin is there. So then what happens? So this has condensed, so nucleus is saved. It's a condensed nucleus, you can say. It's a condensed nucleus. Then what happened? The mitochondria, these are mitochondria. The mitochondria, they start aligning around the flagella in the middle piece. Mitochondria start aligning towards what will be middle piece, Okay? So these are the changes taking place there. And when the changes are taking place, what is happening here? Again, this is your lumen. I'm drawing it a little bit, okay? So right here is, for example, this is one attached to second, attached to third like this. So what happened, that this cell which was here, it start making a membrane like this, so that what happened, that all the cytoplasm is left behind. Okay? All the cytoplasm is left behind, and you have only this kind of like very little cytoplasm, and these sperms will be then released here in the lumen. Okay? And what will be left here? Here will be left like, this thing will be still left there. Only cytoplasm, no nucleus there, right? All the cytoplasm, all the mitochondria has come into the sperm. What is left is only the nucleus. And these are, what is left there, right? These are called residual bodies. Residual bodies. So the residual bodies will be left behind. And that's what we see here. So all the residual bodies are left behind and the sperm is essentially going out. That is making sense? So
The residue bodies are like
whatever left behind cytoplasm there. So the spermatocyte, now these which are formed, now this is essentially the sperm which is formed. So sperms are out, and then you have these residual bodies left behind. So essentially it started with the changes in the cytoplasm. Golgi changed to acrosome vesicle, and then you have the flagellum formation start, then nucleus rotation take place, so the flagellum is in the lumen, and then the chromatin condensation take place, histone is replaced by arginine, and then that lead to nuclear condensation, and then that cause mitochondria, that leads to mitochondrial movement in the kind of middle piece where they will surround the flagella, and then the sperm basically exclude out all the cytoplasm. So all the cytoplasm is left behind and the sperm is released in the lumen, and rest of the cytoplasm is called residual body or the cytoplasmic droplets. Essentially those are the drops of the cytoplasm left behind, right? That is making sense? Any question, any doubt here? Okay, talk to each other for a minute, then you can find out if there is any doubt there.
I mean, maybe it's just that it's getting closer to our limit. You know what I'm saying?
Well, I know that they said, because I know there's some trails that they.
Yeah, kind of makes sense.
They're attached.
Because like, in the drawing, so that happens, then the rotation is like the tail of the vegetable growing into the lumen. And then the nucleus connects, so histones are replaced by proteins, and then these are rich RB, which is about 60%,
Okay, everything is clear here? Any doubt?
So when you say like the nucleus rotates, like it's just actually.
Yeah, it actually rotates. Because beneath is the centriole. When it rotates, along with centriole also rotates. So purpose is the flagellum, which is angling like this, should come into the lumen.
So like right here, the centriole would be like.
Yeah,
over
yeah.
here or something.
Yeah, so right here in your picture, like right here is the centriole. When it rotates, centriole will come here. So that's why it goes like this, right? Any other doubt? You're fine here?
Yeah.
Okay, so that was about the spermatogenesis, essentially in the human, right? Now we are going to talk about oogenesis in mammal. Oogenesis, and when we are talking about this oogenesis, we are specifically talking about the human oogenesis, okay? So, oogenesis is very different from spermatogenesis, okay? It's not as simple as the spermatogenesis was. And there are multiple signals which actually regulate the oogenesis. There can be signals from hormones, signal from paracrine factors, and there are the signals which are specifically related to the tissue anatomy of the ovary, right? So tissue anatomy. So multiple different type of signals are there which are regulating the oogenesis. Now in human embryo, what happened? In human embryo, around 1,000 oogonia. Remember, primordial germ cell, when they reach the sex cord here in the ovary, they are called oogonia on the genital areas. Okay, so those oogonia reaches there, and by first from 2 to 5 months, they divide rapidly. They rapidly divide, okay? And they reach around 7 million. So 1,000 reach, they divide, and they become almost 7 million, okay? And what happened after that, they start dying. So by 2 to 5 months, dividing crazy, and then they start dying. And by birth, around 2 million are left. This is actually probably shown here in this. Here. So they were dividing, they came into, by 5 months they reach up to around 7 million, right? And then they start dying. At birth, you have around 2 millions of them left. And you can see actually their number is decreasing after birth. So what is happening? So when they are there, so whatever surviving, they die, whatever surviving, they immediately undergo meiosis, okay? So surviving oogonia undergo meiosis, and we know why it goes, because you have a very high stride level there, right? And then when they are there, what happened? They undergo meiosis and they reach, so meiosis starts with prophase 1, right? Prophase 1 has stage, what are the stages of prophase 1? Leptotin, zygotin, diplotin, dikinesis, and telophase, right? And then what happened? They don't go through all the prophase. What they do, they reach up to diplotin. So they reach up to diplotin, And when they reach at the diploidine stage, they are arrested at diploidine stage. So they don't go further. Okay? They just stay there. So remember, when the meiosis start, these are no more oogonia. You're going to call them primary oocyte. So oogonia start meiosis, they are now called primary oocyte, right? So primary oocyte, they start meiosis, they reach up to diploidine stage, and they are arrested there, right? So this stage where they're arrested right here, this is called dictyate resting stage. Okay. So now, they stay in this stage for a long time, and then what happened? So at birth, all these primary oocytes are in dictyate stage or the resting stage. Now, at puberty, so say they are arrested in that stage for around 12 to 13 years, right? And puberty, what happened? You have a hormone called luteinizing hormone. This hormone is coming from pituitary. That hormone comes there from pituitary. And what it does, it initiates meiosis. Okay. And meiosis proceed. So first metaphase, first meiotic division complete. First meiotic division complete. That means one cell has given you two cells. Okay. And then second meiotic division starts, and that is arrested in second meiotic metaphase. Second meiotic metaphase. Okay. At this point, ovulation takes place. Okay. And then fertilization initiate completion of second meiotic division. So what is happening? We are saying around 1,000 oogonia reaches there, and developing ovaries, they all divide by up to 5th month. The population reaches up to 7 million, and then they start dying. By birth, the population of the oogonia is almost 2 million, and we are saying whenever they reach there, the surviving oogonia, there are no more oogonia because they immediately start meiosis. So they are called primary oocyte. Primary oocytes start meiosis, and they are arrested in the diploidine stage of the prophase 1. And we call them, they are in dictyate resting stage. They stay in that stage for 12 to 13 years till puberty. At puberty, luteinizing hormone from pituitary come, initiate meiosis, first meiotic division is complete, and then second meiotic division start, but that is stopped at second meiotic metaphase. So they are arrested there. Then ovulation takes place. So this oocyte, which is still primary oocyte, that comes in the fallopian tube or oviduct, and from there the fertilization takes place. When the fertilization takes place, that basically take out this arrest in the second meiotic metaphase, and the second meiotic division is complete. Making sense? Okay, talk about this scenario for a second, then we'll talk about how everything is happening, how all those things are happening.
So, the human embryo is 1,000, and then 2 5 months divides into 7 million, and by birth 2 million, and then undergo meiosis and arrested in prophase 1, as the dictyate resting stage, and then at puberty, the luteinizing hormone initiates meiosis, and then first meiotic division, and then arrested in metaphase 2, and then ovulation, and then fertilization begins again.
The general scheme is clear, right? What is happening? You guys are okay?
Yes.
Okay, now, why and how these things are happening? Now, we are talking about, okay, they are reaching there, they are dying, and then we say the meiosis start. And we are saying meiosis stops, and they are in the dictyate stage, okay? So how that regulation takes place? So we are talking about this thing, what we are saying, meiotic regulation of developing oocyte is regulated by the anatomy. And that anatomy is of what? Because they are in the ovary, so anatomy of the ovary. So if we do a cross section of ovary, for example, this is an ovary, and you do a cross section of ovary. In the ovary, you will find many circular structures like this. Okay. And these structures are called primordial follicles. These are called primordial follicles. Okay. So what happened that they are present in the ovary like this, and they are all present in a stage of dictyate stage. Okay. So if I take out one of this out and draw the structure, how that look? So each of this will look like a structure like this. Okay. This will be surrounded by the cells like this. Okay. Now, and then there will be another type of cells which will be surrounded. Like this. Now, this right here, this is an ovary, or not the ovary, this is an oocyte, primary oocyte, right? And this is, right here is oocyte. And that oocyte is in dictyate stage. This is in dictyate stage. Okay. And the cells which are surrounding, those cells are called granulosa. These cells are called granulosa cells. Okay. And then these cells, these are loosely arranged thecal cells. Loosely arranged thecal cells. Now, what happened that this is how it looks like in the orbit at the time of birth. Okay. Now, what happened all of a sudden, now this is kind of like 12 years like this till puberty, right? All of a sudden, at puberty, there is a hormone called follicular stimulating hormone, FSH. This hormone is, of course, in the pituitary, and this hormone is there. So, all of a sudden, under the influence of this hormone, not all the primordial follicles, not all, a group of primordial follicles, usually they are 5 to 9, almost that is the number, 5 to 9 in that group. What they do, they start growing. They enter a stage of follicular growth. Follicular growth. They enter a stage of follicular growth. Okay? So what is happening? We are saying that FSH is there, the follicle stimulating hormone is there. So you have FSH, and that FSH start follicular growth. Okay? It does two things. Number one, it induces 500-fold increase in size of primary. It is still oocyte, not primary oocyte, of oocyte. Okay? So you have that much size. So what is happening? That this oocyte, which was there, it start increasing in size, 500-fold increase in size. That's the first thing. It also does two things. The second thing is, so I will talk second thing in a minute, okay? So before that, what happens? This is increasing. Now the cells which were here, which were surrounding it, these cells, which were granulosa cells, these granulosa cells granulosa cells, they start secreting GDF9. Now GDF9 is a member of family. GDF9 is a member of TGF beta TGF-beta growth family. Okay. TGF is transforming, it's called transforming growth factors. Transforming growth factors, TGF-beta. Okay. Now these cells, which are granular cell, they start secreting GDF9. And GDF9, it acts as paracrine factor. Acts as a paracrine factor. Paracrine factors are those chemicals which are released from a cell, they affect the adjacent cell, neighboring cell. Okay? So, under that influence, what happened? These granular cells, they start dividing. So they start making their layers like this. So they are concentric layer, more than that. I'm just trying 3, but 5, 6, 7, 8, 9, right? So they divide a lot of those layers. So then this oocyte has grown in size, and these cells which are surrounding it called granulosa cells. Right? And again, the thecal cells are still there. So these cells which are here, these are called thecal cells. Now what happened? I told you the first thing follicle stimulating hormone does, induce 500-fold increase in the size of oocyte. As oocyte is increasing, the surrounding cell also dividing because of GDNF secreted by these cells themselves. Okay? So second thing what follicle stimulating hormone does, it induces expression of luteinizing hormone receptors. Luteinizing hormone receptors. But where? Where it does that? Let me find out a place to write here. On the outermost layer of granulosa cells. So, for example, if this is the outermost layer, right here, they are going to start expressing. So, this will be receptor for luteinizing hormone. So far it is okay? Now, if there are mutations in GDF9, that cause mutation in GDF9 that cause premature ovarian failure. And what that does, and it's too much crowded information, but you're following it, right? What that does, that release two oocytes in oviduct, two oocytes in oviduct. If there are two oocytes in oviduct, that means there are two eggs for fertilization, so that is going to cause the fraternal twins. That's why you get fraternal twins. Otherwise, there's always one ovum or egg is released. But in this case, you can have two, right? So that is the reason you get the fraternal twins, because there is problem in the GDNF9. So far it's okay. Now what happened? this process keep on growing. So they keep on growing. It's okay to erase this? Okay. Now, this thing is growing, and ultimately a fully grown oocyte. How that will look like. So they are growing, they'll keep on growing, right? How that will look like. You will have a oocyte right here, draw it a little bit smaller so that I can draw here. So this will be your oocyte, right? Remember we said that the oocyte, it secrete out extracellular matrix, right? So it will make an extracellular matrix around this. So you will have an extracellular matrix around this. What you call it? What is the name of extracellular matrix around the mammalian egg? Zona pellucida. So you will have a zona pellucida there. And then the cells which are surrounding it, they will also divide like this, like crazy. So you will have more and more cell around this. But right here, you will have an area. I'm just drawing it very simplistic, drawing here. So everywhere, all cells, everywhere are cells. Cells, cells, cells. Right here, there are no cells. Okay? So a cavity will be created. That cavity is called antrum. So a cavity, antrum will be created. And what antrum will have? Antrum will be, antrum will contain a mixture of proteins. So a mixture of different kind of proteins will be in the antrum. And of course there will be hormones. Hormone and other mixture of protein. Now, these cells which are surrounding the ovum, what they will be called, immediately surround, what will we call them? Cumulus cells, right? So all those will be cumulus. And the cell which are next to the zona pellucida, what do you call them? This layer of cell. Corona radiata. Yeah. And then you will have this corona radiata. Now, if somehow what you do, you remove this ovum, or oocyte I will say, you take this oocyte out, only oocyte. Now this is important. If you take only oocyte out, or you take this oocyte plus cumulus. So oocyte or oocyte plus cumulus means you are taking this thing out from here. Now, this mature, this is the mature, right? Now, this fully grown follicle, which is now mature, from this, if you take out just oocyte or you take out oocyte plus cumulus, then immediately start the meiosis. But you need to take them out. But, this is interesting, but if they are here, inside, they will still in the dactyl stage. So that means, what does it mean? It means that inhibitory signal which is telling that you have to stay in the dictyate stage, is coming from these cells, what you call granulosa cells, right? So all those cells are granular cells. These cells become the cumulus cell, and then if you take out oocyte with the cumulus cell, it still go to the next step. It means that the granulosa cell which are surrounding it, those cells contain that inhibitory signal, which tell them like you cannot undergo meiosis. That is making sense? Or no? So talk to each other for a minute, then we'll go further. Then we can basically talk if there's any question. So talk to each other.
Oh.
Between the induces 500
This one. So this is, did he say that?
Is it the beginning thing?
Yeah.
The,
It's like the big one with just the primary follicle cells.
Yeah,
yeah,
I have that.
so it was just the cross section of the,
So I
book.
have that, and then we went to just the primary follicle, and then he says like lutein ring, and then all of a sudden the puberty follicle stimulating hormone countries and then a group of primordial follicles. Yeah.
Starts growing, molecular growth, which induces 5 times increase in size of the oocyte.
Because I only missed it, right? What's 5 now?
It's 5 times the follicles.